One of the proposed mechanisms by which therapeutic anti-tau antibodies reduce pathology in Alzheimer's disease is uptake and clearance of pathological aggregated form of tau by microglia. Here we present a cell-based assay to assess tau uptake by microglia. This assay represents a useful investigational tool to better characterize the mechanism of action of anti-tau antibodies.
On the first day of the experiment, prepare the BV2 cells by first washing them with 1x PBS in the flask in which they have been cultured. Then incubate them with 0.05% trypsin EDTA at 37 degrees Celsius and 5% CO2 until they detach from the flask. Resuspend the cells in culture medium by pipetting up and down 3 to 5 times. Count cells and prepare a cell suspension with a final concentration of 100,000 cells per milliliter in culture medium containing 200 micrograms per milliliter heparin. Add 250 microliters of this cell suspension per well in a 96-well tissue culture flat bottom plate. Incubate the plate at 37 degrees Celsius with 5% CO2 overnight. After thawing previously prepared pH di-tau aggregates on ice, dilute them in 65 microliters per condition of serum-free medium to make a 500 nanomolar solution. Dilute antibodies in 65 microliters of serum-free medium to achieve double the desired concentration. Mix pH di-tau aggregates and antibodies in a 96-well U-bottom plate.